Okay, so we go now to some principle of cellular, let's say physical principle or computational principle, physical and computational. I hope we can hear it. Computational principles of cell navigation, and what I have in mind is neurons in the brain. I think maybe that's better. Yeah, that's what exactly. It's better like this, no? Yeah. And maybe do we have? You can see it is enough. Okay. So here is the question we would like to solve. Suppose, so the question of interest. So suppose you have a cell, and the cell has on its surface, let's say 3, 4, whatever number of receptors. So those are the receptors. And they are diffusing molecules. Molecules move, and they can go and bind here. And when they bind here, as a function of time, you can look, this is time t, this is the receptor that is bound. Let's say bound receptor. And you look if, for example, this one is bound. So if it's one molecule at a time, it can either be bound or 0. 0, 1, 0, 0, 1. This is for this one. And you can integrate over a time of few seconds. If you integrate, if you have a sliding window that is moving, and so you get some kind of curve. If you average, this is average, let's say average over 30 seconds, whatever. So this is for receptor 1. We have receptor 2 and so on, receptor N. And so suppose I give you as a function of time, you have for receptor K, you have these curves. So based on this, how do you move the cell? So suppose given the curves R1 of t, Rn of t, you understand what Rk of t is, or is the average number of receptors, of molecules that have been at receptor K. Averaged number of receptors at receptor 1 at time t. So you give this vector how to move the cell. So given the curves or the vector, how to move a cell. So for this, we can say, for example, that let's make an example together. Suppose you have 2 receptors, and you receive only molecules from this side. Suppose, how do you move the cell? Well, if you move along the gradient, you will move only the cell in that direction. That's easy. If you want to be more specific in how you move the cell, you need more receptors. More you have receptors, and more you'll be refined into how to locate the direction. Right? So if you have something like this, you'll be more precise. If you receive the one that are bounds are mostly that one, you understand that the cell has to go in that direction. Probably the source is going to be in that direction. Okay. So for this, what we defined is a sort of general averaging. So we define A of t. Basically, if you think about how long a receptor stays in here, it's exponential with the rate mu. So a receptor is bound let's say for a Poissonian time, which with mean mu, which is exponential mu t, and this occur at different moment of time. So this is the time. For the receptor 1, it could be at time, for receptor 1, it could be at time, let's say, 2. For receptor 1, it could be at time 3, and so on. So what you can get, and then you want to average over receptors. So you said, I take one of the receptor, an event that occur at time j for receptor I. So, and then, so you sum over all the receptor from j equals 1 to n. N is the number of receptor. Number of receptors. For example, you have here we have 1, 2, 3, 4, 5. So this would be for 1 to 5. And here what we make this a sharp function, a sigmoid function. You take hyperbolic tangent. So this is how we have no justification of this. So this is the hyperbolic tangent. So that you restart your process each time you bind. So here you bind, and you will be bound an exponential time, which is mu. The rate is mu. And then another one binds. So that's a function that measures the average during the time of interest, time t. It measures the number of receptors that are bound. And then we said, okay, how are we going to propel this, that if it has to move? We said that if you have more receptors bound in here, it should move in that direction. So we said that the velocity, v of t, is a velocity v0. Then you look at this function ai of t. So for each receptor I, we look at the position. So this is x1, x2, x3. This is the position of the receptor. x1 is this vector compared to 0. So this is xi normalized. So basically you take the unit vector in this direction, unit vector in that direction, unit vector in that direction, unit vector. So this is x1 or x3 divided by the module of x3. Right, this is just the direction. So you have your direction. For receptor I, you see if receptor I contains or not one of the receptor at time t. So total you sum over the event. And then you normalize it by the total amount of what you have at time t. So this is normalization. So this is how we implemented the velocity. If everything is symmetric, if you have the same amount of receptor everywhere, that is, they are all contain 1, this should be the same everywhere. So if you sum something which is the same everywhere in all directions, that are uniformly distributed, it should be 0. It should not move. And if you have an asymmetry, it will move toward the gradient. So which means that you can xt plus delta t minus xt is v of t delta t. And here, if you want, you can add noise. If you remember the first class, you can add a noise term, d omega. So this is Brownian motion. And this is a deterministic term that this is for sensing the gradient. And this is how we simulated the first graph I showed you. So you have a cell. Then for the cell, you have a certain number of receptors. then you have a source somewhere. This is a source. So the source can produce molecules. The molecule comes, they bind to here, and according to the number at time t of these receptors that are bound, we generate a vector, and the vector moves toward the direction of the source. So what is interesting is that you have the source, you have the cell location, let's say called position P. So this is characterized by a length, L. Sometimes the cell cannot necessarily go in that direction, so it can go in the direction, let's say, this direction, which is characterized by an angle theta. And so you can characterize this angle. You know, we have V, this velocity. You have PS. This is a distance L. This is V. You know, this is a cosine theta. That's this classical formula, where PS is the length L. So this is L. And so you can characterize, for example, the fluctuations of your system going into the right direction. I showed you that because of fluctuation, it doesn't necessarily have a straight line. So this we can characterize this type of motion. Okay, so now suppose whatever you agree or you disagree with the elementary model, how would you add on this model to account for multiple gradients? So question, this is the model, and so now the next question we can ask is how you complexify this model. So accounting, so this is B, A was this question of interest and this first attraction motion. This is attraction motion. B, what happened if you want to account for multiple gradients? So I'll show it to you. The cell needs to account for multiple gradients for many reasons. For example, the cell starts here. It should go through a corridor, as we said. So the cell basically has to go from this location. It has to arrive to this point. And then, if it has been attracted to that point, it should in some sense forget about the fact that it has been attracted here to move in that direction, and then finally move again from this point, let's say, to this point, which should be the final destination. This is about being attracted from here to here. But how to go from this point to that point? How shall we modify the model? For example, and this is the interesting part, is that maybe there are receptors or co-receptors that can be bound to the two types of morphogen. Let's say the blue one is the attraction. This is an attractant, A. And the green one can either be an attractant, A2, something that attracts, or a repellent, 2. Which means that if A1 is bound, you have a certain strength to go in that direction. If A1 and A2 are bound, the attraction is enlarged, is increased. This is called an amplification. But if A1 and the repellent are together, maybe it annihilates. And then there is no motion. And if the repellent is activated alone, it it's going in the opposite direction. Okay, so there are different cases. Repellent alone, go in the opposite direction. Now the cases. So let's do the cases. On your computer, you have cases. Repellent alone, 1, opposite direction. Attractant alone, it goes in the direction, the right direction, correct direction, whatever correct means. 3, if you have two attractants, A1 and A2, which means we have two attractants, then it amplify in one direction. And if you have attractant plus repellent, maybe nothing happens. It annihilates. Nothing. Nothing happens. Okay, now we need to formulate this in mathematical terms. Now we have put a voice, an understanding of what we want. How shall we modify this? If we say that this A is an attractant, now suppose I have two independent molecules. I can generate another BA or whatever with maybe a different time scale, mu. I can say it has a different time scale or not. And look at this term, and suppose I have two attractants. Okay, I'll do two attractants, and then at home you can do the... So let me erase this part. Now suppose you have two attractants. How would you modify the velocity? You can say the velocity is equal to V0, it doesn't change. If you have one attractant at a time, or one repulsant at a time, you can say, look, one can act, and the other can act, one independent of the other. So I need to have, let's say, two terms. So I can have a term alpha 1. Okay, I write everything. A, I call A, the one that I have here, A of t, xi, divided by xi, this is what we said, divided by the sum of ai. This is for repellent 1, or attractant 1. If it's also a repellent or an attractant, I say I have another one, which is called bi of t, xi divided by xi, divided by the sum of bi. And for bi, I can take the same thing. If there are two with the same weight, alpha 1 equals alpha 2, if B1 is a if this is a repellent, what is the sign of alpha? It's going to be minus. So if this is positive, it's an attractant. If it is negative, it is a repellent. Okay, so if this is 0, the repellent can do the job. If this is 0, the attractant will do the job. And now we need to formulate the possible coupling, the possible interaction. So now there is a term, call it, I don't know, plus alpha 1, 2. And now we need to see, for example, if they are the two together on the same receptor, which means AIT multiplied by here. If they are interacting, how we, in mathematics, how you describe the interaction of two terms? You do multiplication, that's right. So AI, for example, BI, and then you do the same, XI divided by this, divided by the sum of AI, T, BI, T. where AI and BI satisfy the same rules that is defined here. So if this is when they are going in the right direction, when AI and BI are bound, suppose alpha 2 is positive, this could be, for example, an amplification. If this is much bigger than each of alpha 1 and alpha 2, let me erase the second part here. Then you have an amplification.
What is X again?
I'm sorry?
What is X again?
X?
Yes.
Wait just 1 second. So, again, you have a cell. This is the center of the cell, and I put a receptor compared to the center. I put a receptor at position x1. I put another one at reception at position x2. So this is this vector.
Oh yeah, so A, B is at the same position. Oh yeah.
Suppose they have the same position. They are bound. So this is for receptor A, and this is for receptor B. It's the same. I put two sides.
Yes. So, yeah.
A and B.
Got it.
This is for A, and this is for B. And the same. They have one side, A and B.
Yeah, so they have different positions. Yeah.
Yeah, the same position.
Okay.
And so you can have an amplification if alpha 1, 2. Alpha 1, 2 is much bigger than an alpha 1 or alpha 2. Okay?
Yeah, sure, thank you.
And also what you can have is the opposite, when you have AI and B. AI is bound, and BI is not bound, and this has an effect. And then in that case, what you need is to replace BI by 1 minus BI. Okay, I think I give you a lot of information. Why don't we stop here in terms of the complexity? So with this type of parameter, and I'll show you that you can generate a cell that moves according to attractant, repellent. When it arrives to the repellent according to the parameter, it can be deviated and move toward another attractant, and so on. So because for many years we've tried to find minimum rules that will allow to understand, because to that day the problem scientifically is not solved. We don't understand exactly how cells are moving toward the right place and position and stopped. We started this probably 15 years ago when I started working with Prochian at Collège de France. And the field has moved not so much in that direction. From a biological point of view, it moved a lot in the identification of the molecule. Which molecule is involved into recognizing the specific morphogenes and how they could possibly interact. This has been working hypothesis, and a lot of work has been done in that direction. So now, how do we use this to construct the path of a neuron, of the axon? So for example, if you call now those are the rules for each time t. So now constructing a path. This is C constructing a path. Suppose you start here and you have a source here. First of all, you have to say when the cell stops. You can, now we have to define something that is not in the model here. So you can say, if this is expressed in a gradient, as I explained to you before, so you have a source that emits molecule, and this guy is going to be sensitive to this molecule, so it's moving in that direction. And we can say, for example, so I impose a rule that if all the receptors are more or less all the time saturated, they contain the molecules, then you stop. This means that you have to say that all the AI, for some then there's going to be a position, a time, which is going to be bigger than a certain threshold for all I. And then you say, okay, it means that they're all saturated, it means they're very close to the source is moving molecules all around, and if you have this, then you can say tell the computer to stop moving. There's something we have to impose. This is one. Another way to look at it is to say that, for example, if none of them, if all A, they are very small, so I put another threshold, let's say small, it means it receives nothing. And if it is nothing, so this means that nothing is received. And then you stop. Okay. So I want to do one more thing in this direction. Is the following example that we saw from the beginning. Can I erase this part? I want to construct, you remember the first example I showed to you, going from the retina to the back of the brain, to the tectum or to the visual cortex. We had the retina, so this is application, A, B, C, D. So suppose you have neurons that start at different positions from nasal to temporal. And I showed to you that if you go to the cortex, if you go to the tectum, so they project in the brain, and this one will stop here. This one will stop here. So that's a bit more tricky. Before we said you stop when you're near a source. That's okay. It's not that complicated. Here we have reached a second layer of complexity. How would you achieve such a mapping? Suppose you have a source. Okay, you have a source somewhere, which means that you can have a gradient here. Suppose we start with this. But what is this enough to have this? So suppose we have one gradient. Is this enough to have such a patterning? Obviously not, because you see that if you start perpendicularly, it plays a role. So you need to be in dimension 2. Okay, now we have understood we need to be in dimension 2. How do you implement a dimension 2 graph pattern? How do you implement it into the model? So who has an idea? Here we need a new ID. So this is to convert a question in biology into computation. So what is the new ID that you need? You say, before I explained that how you can sense a gradient and stop at the source or stop at the very beginning, whether you are too saturated, too much, or too little. But how do you stop at intermediate position? That's not easy. It took us time to think about it. Don't you think? Don't think that it took me a minute to solve the problem. So we said this. We said, since we can play only with the parameter that we have, on which parameter can we play? Each receptor, they are produced the same. They have the same characteristics. So we cannot play on the mu parameter, for example. You remember mu was the rate at which the time they stay, the bound of the two molecules, the receptor between the molecule, the time they stay together. Because if they are expressed genetically, they should be the same. So we cannot play on mu. But what we can play on
Alpha.
Is what?
Alpha.
Yeah, okay, alpha. Alpha is abstract. Alpha is let's say, when you have already here some kind of competition. Now suppose we have only one, so alpha is 1. What are we left with? With one parameter, which is what? What? We're left with one parameter, which is not written on the board, which I've written on the previous one, is the number of receptor. I can play with the number of receptors. What does it mean?
Threshold. How sensitive?
So that's right. So now suppose I have only 2 receptors, and suppose this one has many receptors. So to saturate this one, maybe you need to be, it should be the opposite. To saturate this one, to have everything full, what do you need? Compared to that one, if you have 2, maybe for this one, you don't need to go that, no, I was right. I don't need to go that far to have only 2 saturated. But if I have many of those, many receptors here, maybe you have to be much further away in order for them to be saturated with the same rules. Because maybe here you need, 20 bound receptors, so you need to be closer to the source compared to this one where you need 2 in average. And you can show that if you do this, you have a patterning, and this was justified by the genetics, which means that you can have a graded expression of receptors to start with. Okay? So the idea is that if you have few amount of receptors, the cell needs to travel less compared to the one with many more. All right, this is an hypothesis. I cannot prove this, but at least I converted for you a situation where you want to label. We started with going through a single source. I explained how to build the case where you can have a repeller and an attractor. And then we said the third layer of understanding is when you need to have a graded expression. Of course, it's not like this, when you have neurons, they have usually glial cells, you have a bundle of many neurons, axons going together. It's more complicated, but having rules, elementary rules, is quite important because it helps to understand what's the complexity by which cells move inside or find a target. For example, I'll go back now to the presentation, then we'll make a break, and then we will go to the notion of sensing. All right, so let's go back